Hello students, how are you? Welcome back. Students, today's class is going to be super interesting because I am going to conduct an experiment to understand the concept of cell wall. So today's topic is cell wall, and watch this video up to the end without skipping. I am going to perform an experiment which is going to be very interesting. Now let's continue. Cell wall. What is a cell wall? It is non-living rigid structure that gives shape and protects the cell from mechanical damage and infection. So yesterday we have discussed about plant cell. So what was that rigid layer that surrounded the plant cell? It was cell wall. What is the function of cell wall, students? It gives support to the cell, structural support to the cell, and also it gives a proper shape to the cell, and also it prevents the bursting of the cell because of excessive pressure. Okay, suppose, imagine cell does not have a cell wall. There is only a layer called as plasma membrane as a delimiting membrane. A plasma membrane is a flexible, selectively permeable membrane. Okay, under the plasma membrane we can see cytoplasm liquid, there is nucleus, there is vacuole, and many cell organelles. Suppose if water molecules start entering into the cell through the plasma membrane by process called osmosis. When water molecules keep on entering into the cell, then what happens? The cell inflates, it swells. At one point, the plasma membrane couldn't bear that excessive pressure inside the cell. All of a sudden, the plasma membrane is going to burst, it is going to rupture. It is going to burst. When the plasma membrane bursts, then what happens? All the inner contents come out of the cell. Then eventually the cell is going to die, isn't it? So this is what is going to happen when cell wall is absent. Then you may ask, sir, but animal cell does not have cell wall. Is animal cell is going to burst? No. Animal cell have several mechanisms to protect from the bursting. So that is, one of the mechanism is osmoregulation. In the animal cell there is a cytoskeleton which gives a structural support from inside. So by this type of mechanisms, the animal cell will protect itself from bursting in spite of absence of cell wall. Now let's see the experiment. Students, I have made a polythene cover balloon. So I have sealed this polythene cover, and by using this straw, I am going to inflate this polythene cover. Imagine that this is a plasma membrane. Okay. I am inserting the straw into this polythene bag. I have inserted the straw into the polythene bag. Imagine that this is the plasma membrane. Plasma membrane. Is it getting inflated? Watch. Yes, air is entering. Yes, now our cell is ready. So imagine that this is a cell. The cell is covered by plasma membrane. The cell is covered by plasma membrane which is flexible, selectively permeable membrane. You can see here it is flexible. Okay. What are the different components present in the plasma membrane? Lipids, carbohydrates, and proteins. These three components will be present. Now imagine that inside this cell there is a cytoplasm, nucleus, all cell organelles are present in this under this plasma membrane in this cell. Now imagine what happens during excessive entry of water molecules. When water molecules enter too much, then the cell is going to swell. Okay, at one point the plasma membrane could not tolerate the pressure and it can burst, simply it can burst, releasing all the contents. Then what if there is a rigid layer which is going to protect this plasma membrane? That rigid layer is going to be our plastic container here. Imagine this is the cell wall. I am putting the cell inside the cell wall. Now my cell is safe inside the cell wall. I am covering it with lid. Now our cell is protected from all the sides. Okay. Even though the cell swells, the cell wall will not allow the bursting of the cell. Okay. Suppose if I try to hit the cell will not burst because the cell wall is a rigid layer. Okay. It will protect the cell inside. The cell will be safe inside the cell wall. Okay. I try to hit this from all the sides. The cell is not going to burst. So this is the protection which the cell wall provides to the cell students. This type of rigid cell wall we can see in the plant cells and also in the fungal cells. Okay. So now I am removing the cell wall. So this is the cell without cell wall. We can also call it as protoplast. Suppose imagine that the water has entered into the cell. Okay, now it is completely inflated, and at one point the plasma membrane cannot tolerate that pressure. What happens? The cell is going to rupture. The plasma membrane bursts, releasing all the contents to outside. The cytoplasm, the nucleus, all the cell organelles will come out and the cell is going to die. So this is what is going to happen when cell wall is absent. Understood, students? Understood the concept? If you like my efforts, then give a big like to this video. Students, now let us discuss about the concept of endomembrane system. What is an endomembrane system? Endo means inside. Inside the cell there is a membrane system called endomembrane system. The four cell organelles, namely endoplasmic reticulum, Golgi complex, lysosomes and vacuoles, these four cell organelles, they form endomembrane system. So why only these four cell organelles? Because these four cell organelles, they work in a coordinated fashion. That's why they form endomembrane system. First let us discuss endoplasmic reticulum. What is endoplasmic reticulum? Endoplasmic reticulum is fine tubular network which is scattered inside the cell in the cytoplasm. We call this cell organelle as endoplasmic reticulum. Okay, if you observe inside the cell there is a fine tubular network which is scattered throughout the intracellular space. This network of fine tubular structures is called endoplasmic reticulum. Now, a small portion, when it is expanded, when it is enlarged, it appears like this. This is obviously the nucleus. In the nucleus is nucleolus. If you observe here, this is the cell organelle endoplasmic reticulum, and even this is also endoplasmic reticulum. It is a network of fine tubular structures which is continuous with the outer surface of nuclear envelope. Okay, so now the endoplasmic reticulum, the fine tubular network, it divides the space inside the cell into two compartments. Okay, what are the two compartments? The space which is enclosed inside the ER is called as luminal compartment. Luminal compartment. So what is luminal compartment? The space which is present inside the endoplasmic reticulum is called luminal compartment. Whereas the space which is outside the ER, this region, it is called cytoplasmic compartment or extraluminal compartment. Understood, students? What are the two compartments? Luminal compartment inside the ER, cytoplasmic compartment or extraluminal compartment which is outside the ER. Now, endoplasmic reticulum is of two types. The ER surface showing the ATS ribosomes, granular structures called ATS ribosomes, then that ER is called rough endoplasmic reticulum. What is the function of RER? RER is involved in protein synthesis. What is its function? It is involved in protein synthesis. With the help of these ribosomes, the endoplasmic reticulum can involve in the process of protein synthesis. Next, what is SER? What is smooth endoplasmic reticulum? The ER which does not have any ribosomes attached to it is called smooth endoplasmic reticulum. And what are the functions? It is involved in lipid synthesis. And in animal cells, the SER is also involved in synthesis of lipid-like steroid hormones only in the animal cells. So this is about endoplasmic reticulum. Understood, students? Now let us discuss the next cell organelle, Golgi complex. Golgi complex. The Golgi complex, it is closely associated with endoplasmic reticulum. The Golgi complex shows several flat disc-shaped sac-like structures which are arranged parallel to each other. So this is a network of flat sac-like structures. They are arranged parallel to each other. So this cell organelle, it is closely present near the endoplasmic reticulum. This cell organelle was first discovered by a scientist called Camillo Golgi. Discovered Golgi complex cell organelle. Okay, so here in the Golgi complex you can see a flat disc-shaped sac-like structure. A single flat disc-shaped sac-like structure is called cisternum, and many are called as cisternae. Several cisternae are arranged parallel to each other. You can see that there are two phases in the Golgi complex. The phase which is having a convex shape, this is the convex shape. So this phase is called as the cis phase. What it is called? The cis phase. It is called as the cis phase. Whereas the other side, where you can see a concave surface, so this phase is called as trans phase. It is called the trans phase. Understood, students? So here in cis phase, new cisternae are formed. How new cisternae are formed? Let us see. The endoplasmic reticulum, ER, it will pack the proteins in spherical structures which are called as vesicles. The vesicles contain unmodified proteins. So this vesicles which enclose the unmodified proteins, they detach from the endoplasmic reticulum and they move slowly towards the Golgi complex. The vesicles eventually they fuse together and give rise to young cisternae near the cis phase. That's why the cis phase is also called as formation phase. It is called the formation phase. Why? Because the new cisternae are formed in this convex region. So what is the shape? Of cis phase? It is convex. So new cisternae are formed here in the cis phase which is in convex shape. That's why it is called formation phase. Next, so when the vesicles fuse with the cisternae here, the protein will be delivered into the Golgi complex. Now this protein will undergo several changes which are called as modifications. Eventually, the protein gets modified into a functional protein inside the Golgi complex. As the new cisternae are formed, the old cisternae will be pushed towards the trans phase. The old cisternae will be pushed towards the trans phase, and here in the trans phase we can see the matured cisternae. So this is a matured cisternum, that's why it is also called as maturation phase. Maturation phase. What is the shape? The trans phase is having convex shape. It is having it is having concave shape. This is the concave shape. It is having the concave shape. Understood, students? So this is about the cis phase and trans phase of Golgi complex. So what are the functions of Golgi complex? First one, it is involved in modification of proteins. Modification of proteins. This is one of the functions. And it is also involved in glycosylation, a process called as glycosylation of lipids and proteins. What is glycosylation? The process in which the sugar molecules are added with the lipids or proteins is called as glycosylation. Okay. Next, another function is packing. Packing of materials. Packing of materials. Golgi complex also packs the different materials which need to be transported to different destinations inside the cell. Once the material is thoroughly packed, then what happens? The packed material will be delivered to outside in the form of a vesicle. The vesicle detaches and it will transport that material to different places inside the cell. Okay, not only the material will be transported inside the cell, but also the material can be also sent out of the cell. We call that process as secretion. What is that process called? Secretion. Golgi complex is also involved in secretion of different materials. That means the packed materials are sent outside the cell. Understood, students? Golgi complex has one more function, that is it also forms lysosomes. It also forms lysosomes, cell organelles called as lysosomes. So copy down this, students. Students, now we discuss lysosomes. Lysosomes. Just now we have discussed that Golgi complex, it also gives rise to cell organelles called as lysosomes. What are lysosomes? Lysosomes word is derived from a word called lysis. Lysis means breakdown. Lysis means breakdown. Lysosomes are membrane bound cell organelles. They are covered by a unit membrane, and inside the lysosomes there will be enzymes called acid hydrolases. Acid hydrolases. Okay. These acid hydrolases, they are involved in lysis. Lysis means breakdown. These acid hydrolases, they can break down the old cell organelles which are present inside the cell. That's why they are called lysosomes. And what are the different types of acid hydrolases present inside the lysosome? That includes proteases. Proteases will hydrolyze the proteins. Carbohydrases. These are the enzymes which will break down the carbohydrates. Lipases. These are the enzymes which will hydrolyze the lipids. So these are the three types of enzymes which are called acid hydrolases. So when these lysosomes, when they rupture, then what happens? They will release the contents to outside. The acid hydrolases, when they come to outside, the acid hydrolases, they will break down the old cell organelles and dissolve them. Okay. So in this manner, the old worn out cell organelles will be recycled inside the cell. This is the function of lysosomes. So what is the function? Lysosomes break down old worn out cell organelles. Old worn out cell organelles. This is the function of lysosomes. They are present in plant cells as well as in animal cells. Now the last cell organelle of endomembrane system, that is vacuoles. Vacuoles. So let me clear some space for vacuoles. Okay, vacuoles. So what are vacuoles? Vacuoles are also a membrane bound cell organelle. It is covered by, the cell organelle is covered by a membrane. The membrane is called tonoplast. What is the membrane called? Tonoplast. The membrane which covers the vacuole is called tonoplast. So what is the function of a vacuole in the plant cell? In the plant cell, the vacuole occupies 90% space in plant cell. So because of such a large sized vacuole, all other cell organelles will be pushed to one side, including the nucleus. Okay, so the largest cell organelle in the plant cell is vacuole. It occupies 90% space inside the plant cell. And why it is so large? Because the vacuole stores water. It also stores a material called sap. It also stores several waste materials of the plant cell. Waste materials. And also it will store several mineral ions. So that's why its size is large. The membrane called as tonoplast, it is having one function, that is the tonoplast, by using ATP energy, it can pump more ions, mineral ions into the vacuole. So this is one of the important properties of tonoplast. So because of the pumping activity of mineral ions, there will be always high concentration of mineral ions inside the vacuole. Okay, next. In addition to a plant vacuole, this is a plant vacuole. Other vacuoles are also found in unicellular organisms. In kingdom Protista, if you observe amoeba, in amoeba there is a vacuole called as contractile vacuole. What is that vacuole called? Contractile vacuole. So what is the function of this contractile vacuole? The contractile vacuole has a function of excretion. It is having the function of excretion. So all the unwanted water which is present inside the cell, it will be sent out. That is the function of contractile vacuole in amoeba. And many protists such as the amoeba, paramecium, they have another cell organelle inside them called as the food vacuole. Food vacuole. Food vacuole is present in unicellular organisms like amoeba, paramecium. What is the function of this food vacuole? The food vacuole, it will enclose the food material, and the digestion of food material takes place inside the cell organelle. So that's why the vacuole is called as food vacuole. So this marks the end of the endomembrane system, students. Copy down.